I'm John Ramsey. I'm studying for a PhD in mixing viscoelastic fluids in process equipment. I did my undergraduate degree here and I'm currently midway through my PhD. We did 3 experiments. We did some particle image velocimetry, or PIV, planar laser-induced fluorescence, or PLIF, and some rheometry. The aim of each experiment is to sort of get to grips with fluid dynamics within process equipment and gain a better understanding of how processes work. Principles of particle image velocimetry it's a method to view flows within vessels, and it works a little like a speed camera. You take 2 images and track the location of a particle, and by knowing how far it's moved over a set time, you can work out the speed at that point. There's a laser synchronized to a computer and to the camera, and this fires 2 pulses. It takes 2 pictures, and through tracking where different particles are, we get a full velocity field for the vessel. And this allows us to calculate local velocities, but also other things like local shear rates, vorticities, sort of local turbulent effects. Now, this technique's really useful in that it allows us to see within vessels themselves and see how things actually behave. This is really useful when trying to optimize processes. So in industry, one may have dead zones within their vessel, and this technique allows us to see where they are, where bypassing occurs. And also from a design point of view, PIV is very useful for validating computational fluid dynamics data, or CFD, which is a computer prediction of a design. And so working in parallel with experimental and simulation techniques, we can be sure that before we go out and build a full-scale example of our industrial equipment, we know ahead of time that it will work and it is the most efficient design for the application. So PIV involves having a geometry, such as a stirred tank with an impeller that we shine a laser plane through. Now, first we have to put our geometry inside a square-based vessel. So this is to sort of minimize any laser attenuation that we may get to keep the light sheet in a thin, narrow beam. What we then do is add a small amount of reflective particles to the flow. Now, when the laser beam hits these particles, they'll reflect and come up as bright spots on any image, and this allows us to track their location. From there, we then fire the laser, take a sequence of photographs, which we can then pair off with each other, and over time we can calculate the velocities at each individual location. This gives us a vector map of fluid velocities. From here we can do further processing to look at the magnitude of these velocities, the direction they're going in, and calculate additional parameters such as shear rates and vorticities, which are very useful to know for industrial equipment. Planar laser-induced fluorescence, or PLIF, is very similar to PIV in that you still fire a laser through your geometry, but this time, instead of tracking reflective particles, you track the location of a fluorescent dye. Now, first we have to attach a cut-off filter to the camera lens, which eliminates any stray reflections, which were beneficial for PIV, but in this case will detract from the overall image. Then add a fluorescent dye, in this case rhodamine, which fluoresces at a slightly different wavelength to that of the laser light that's put in, and so this will fluoresce bright pink when green light is applied to it. So from there, we then inject this into the vessel, in this case agitated by an impeller, and by looking at the local brightness of the dye and on the process data at the local grayscale values, we can work out local dye concentrations, and we can use this as a substitute for knowing, say, a reactant concentration if a reaction is important, or when trying to mix two fluids we need to know local concentrations to ensure homogeneity. And this is really useful to see cavern formation in non-Newtonian fluids over time, which is something that is undesirable in a lot of process equipment. We perform rheological characterization on materials in order to understand how they behave when subjected to shear. So we use a piece of equipment called a rheometer, which consists of a geometry, in this case a cone and plate, and a base plate. And by loading the material in a gap between these two surfaces and controlling the rate of rotation of the top plate, we can calculate shear stresses, and from that we can calculate material viscosities, which we can use to then work out Reynolds numbers within our process. So first, because the machine operates at micron tolerances, we have to zero the gap, which just involves lowering the head until the two pieces touch. From there we load our sample into the open gap, and ensuring that we don't either overfill or underfill the geometry, as these are carefully calibrated for the exact amount of fluid. We then begin the experiment where we vary the shear rate by controlling how fast the cone and plate geometry rotates, and we measure the torque, which gives us the shear stress. By working out the gradient between these, we get an apparent viscosity, and by monitoring this over a range of shear rates, we can see how the material viscosity changes over time. And this is really important to know in our process. As we can see here, we've got our material, which is carboxymethyl cellulose in water, or CMC for short, a dilute polymer solution, exhibiting shear thinning behaviour with no yield stress. And so in this case we can assume it's a power law fluid, and so if we were operating in a stirred tank we could use something like a Metzner-Otto correlation to work out average shear rates for the tank. The fundamental principles underlying all the experiments I do are ultimately fluid dynamic based. So a lot of fluid flow, a lot of liquid mixing, and a lot of rheology, and it's tying all three together that really gives these experiments value. And so it's really useful for realising that issues aren't kept separate in reality, they all tie together, and understanding one aids your understanding of another.